Welcome senior one to a new biology session. Today, inshallah, we'll continue discussion about inheritance, interaction of genes. We have here start talking about lack of dominance. In this type of inheritance, we studied before the inheritance in the plant called antennum plant, in which there is two colors, red and white, but when they are pure and crossed together, the result will be 100% a new trait, which is purple. So there is no dominant gene here, and there is no recessive gene. The two genes, the red gene and the white gene, express themselves, and the new trait appear intermediate between the two traits of the parents. So we say that it is lack of dominance. There is no dominant gene. In the human, there is a studying of lack of dominance, like the studying of blood group, inheritance of blood group. In the human, we have 4 blood groups: A, B, AB, and O. A have 2 genotypes, AA and AO. B have 2 genotypes, BB and BO. We can discover here that A can be pure and can be hybrid. This means the gene A is dominant over O. And the gene B is also can be pure and can be hybrid. So the gene B is dominant over O. If A and B come together, a new group formed, which is AB. So we can discover here, we can say that A and B are not, there is no dominance between A and B. There is lack of dominance between A and B. Every one of them have the same power, have the same influence in appearing a new trait. And finally, the blood group O appears only when the 2 genes are O, when it is pure. So we have, in studying the blood group, we have there is a complete dominance between A and O and B and O, like Mendel. And we have lack of dominance between A and B. And we have another way of studying of inheritance, which is multiple alleles. The meaning of multiple alleles, there is 3 allelomorphic genes. We have 3 genes, which are A, B, and O. Not 2 genes only, but the individual carry 2 genes only. 1 pair of these genes, one from the mother gamete and one from the father gamete. But there is 3 alleles, 3 genes in the inheritance of blood group. The complete dominance, yes, we have complete dominance between A and O, B and O. And we also have lack of dominance between A and B. They are equal in power. No one of them is dominant over the other one. So if they come together, a new trait appear, which is blood group AB. By inheritance of blood group, we can see this example. If 2 parents, male parent has blood group AB, and the female parent has blood group O. If they are married together, the gametes of AB may be A and may be B. But the gametes of O, all of them is O. The offspring, the new individuals, the kids of this marriage, some of them will be AO, which is blood group A, and some of them will be BO, which is blood group B. So there is no one of them O, and no one of the children are AB, and no one of the children is O. So this is a new way of inheritance, which is multiple allele and lack of dominance. Here we have study, we come to another part of the lesson. We have to study chemical classification of blood groups. Chemical classification of blood groups. This study is important to know who can donate blood and who can receive blood. Not any group can give any group, and not any group can receive from any group. There must be some rules in studying how to donate blood how to receive blood. It will be very dangerous if someone receive blood from a donor with a blood group which is not compatible with his blood group. For example, if someone has blood group A and the donor with blood group B gave him blood, it will be big dangers because blood group A is not compatible with blood group B. If a person has blood group A, you cannot give him blood from another one has blood group B. He will die. Why will die? Because there is some chemical substances in the blood we'll talk about called the antigens, and another chemical substances called the antibodies. When they are found together, they can cause agglutination of blood. They can cause the chemical reaction which causes the death of the individual. Let's talk about antigens and the antibodies. You know the blood group, if we have a drop of blood like this, the blood contains many cells. Some of them are red blood cells. Some of them are red blood cells like this. Red blood cells. The blood have red blood cells like this. They are red blood cells. And also we have white blood cells. We have also white blood cells. White blood cells WBCs and red blood cells RBCs. The blood contains white blood cells and red blood cells. And also there is something called platelets. Platelets here, they are not a complete cell, but they are important in clotting of blood, making the injury covered. The blood clotting is covering the injury tissue to heal. So we have red blood cells, we have white blood cells, and we have platelets. And finally, we have the liquid of the blood, which is called plasma. The liquid of the blood, which is called plasma. So there is many components of the blood. There are many components of the blood. You can say there are 4 components: red blood cells, white blood cells, platelets, and the plasma. And there is chemical substances called the antigens. Chemical substances called antigens found on the red blood cells surface. They are found on the surface of red blood cells like this. Like this. The antigen here, and the antigen here, antigen here, antigen here. On the surface of the red blood cell. Antigens, we have 2 types of antigens. Antigen A and antigen B. We have 2 types of antigens. Antigen A and antigen B. Blood group A has antigen A. Blood group B has antigen B. Blood group AB has antigen A and the antigen B. So, if this person has blood group A, if his blood group A so he has antigen A. He has antigen A. Because he has these antigens on the surface of the red blood cell. And he has also antibodies, chemical substances called the antibodies. These chemical substances are not found on the surface of the red blood cells, but the antibodies are found in the plasma, in the liquid of the blood, which is called plasma. So we have here antibodies, antibody A and antibody B. Who has antibody A and who has antibody B? The one who has antibody A, opposite to the group, the name of the group. So if you have blood group A, you have antigen A, and the antibody B, you have anti-B, antibody B. So the person with blood group A has antigen A and the antibody B. So there is no chemical reaction between them. Antigen A and the antibody B, there is no chemical reaction between them because they are different. Antigen different from the antibody. This found in the blood of one has blood group A. Type. Someone has blood group B. Has what? Has antigen B and the antibody A. Anti-A. Somebody has blood group AB, has antigen A and the antigen B, and no antibodies at all. In the plasma, there is no antibodies at all. So AB can receive blood from any other person because the chemical reaction happened because of the antibody. The antibody makes a chemical reaction if the same type or the antigen has the same name. Yani the reaction happens if anti-B comes with antigen B. So if somebody has blood group A and you give him a blood or B, a person with blood group B, so the person with blood group A has antigen A, has antibody B, and you give him blood group B with antigen B, there is a chemical reaction happen between antibody B and the antigen B, and the blood agglutination happen. The blood destroyed, the blood cells are destroyed, and the person can die. So anti-B doesn't come with a antigen B, and the anti-A doesn't come with antigen A. So this person with blood group AB has antigen A and antigen B, but he doesn't have any antibodies, so he can receive blood from any other person. The one who has blood group O has no antigens at all, but has two types of antibodies, antibody A and the antibody B. This person can donate blood for anybody, can donate blood for any person because he doesn't have any antigens, but he cannot receive blood from anyone. He receives blood only from blood group O. So the blood group AB can receive blood from anybody. Why? It doesn't contain antibodies. It doesn't contain antibodies. So it can receive blood from any person, from any blood group. But blood group O has the two types of antibodies. You cannot give him blood group A. You cannot give him blood group B. You cannot give him blood group AB. But you can give him only blood group O. And you can donate your blood. The one with blood group O can donate his blood for anyone because he doesn't have any antigens. So we say that blood group O is called universal donor, and the blood group AB is called universal recipient. Universal recipient. So who can give blood to who? It is very important to know who can give blood to who. Blood group A can donate blood group A and the blood group AB. Blood group A can donate blood to a person with blood group A and the person with blood group AB. Blood group B can donate blood to a person with blood group B, the same, and they can give a person with blood group AB. AB receive from anybody. Blood group AB can receive blood from any blood group, but donate only itself. Can receive from any blood group, but donate itself only. And the blood group O can give blood to any blood group because it is a universal donor and receive blood from O only, like this. You can look at the description here. Blood group AB is called what? Universal recipient. It can receive blood from B. It can receive blood from O. It can receive blood from A. And also it can receive from itself. Can it donate any blood group? No, it can donate itself only. Donate AB only. Type blood group O is a universal donor. It can give blood to B. It can give blood to AB. It can give blood to A. and it can give itself, but it receives blood from itself only. Receive blood from itself only. Type blood group B can donate AB and donate B. Type can receive blood from O and they can receive blood from B. Blood group A can donate blood to A to itself and can donate blood to AB, universal recipient. And it can receive blood from O and it can receive blood from itself. So we have to remember which blood group is a universal donor, which is O, and which blood group is a universal recipient, which is AB. O is a universal donor because it doesn't have any antigens. AB is a universal recipient because it doesn't have any antibodies. Is it okay? We have a problem here. A man with blood group AB was married a woman with blood group O. We studied this before. What are the expected blood group for their children? Any children can be O or AB, or the result will be, what is the result will be for the children's? Some children's will be A and some children's will be B. No one of them will be O and no one will be AB at all. And this is used for solving some problems and judgment about the parental issues. Antigens are found on the surface of the blood group. Antigens are found on the surface of the blood group or the red blood cells, the surface of the red blood cells. Antibodies are antithetic to antigens. They react with antigens and found in the plasma, the blood plasma. Risk of blood transfusion. The risk of blood transfusion. You cannot give blood to anybody. When the transfused blood to a person incompatible with his blood group, some symptoms appear on him, such as headache, chest pain, and these symptoms often end with death. If you give him blood different group, if someone is A and you give him blood group AB or give him blood group B, it is very dangerous. Viral infection can be transferred to the receiver as hepatitis and AIDS. So you have to check the blood group and you have to check the blood before donation. How to determine the blood group type? How to determine the blood group type? The test, if you go to know your blood group, what is the test happen? They make a test for you to know your blood group. What is the test? How can they make the test? They bring a slide like this. They bring a slide like this. And they put on it 2 drops, one of them antibody A, and the other one is antibody B. This blood group, antibody A, and this blood drop, this drop is antibody B. And they bring the blood of the person which we want to know his blood. And we put the drop of blood over the anti-A, and we put the drop of blood over anti-B. What will happen? Agglutination will happen in anti-A only if the blood group is A. In the school book is better. It is clear, more clear in the class, the student group here. Here. If you have agglutination of blood with anti-A, so this blood group is A. Agglutination happen with A. This blood group contain antigen A, so it is the blood group A. If the agglutination happen with anti-B only, the person will be having blood group B. If agglutination happen with two the drops, anti-A and anti-B, so the person has antigen A and antigen B, so the person has blood group AB. If no agglutination happen, so the person doesn't have any antigens, so it will be having blood group AO. So if agglutination happens, the blood destroyed and frozen or maybe solidified like this, with A, anti-A, it is blood group A. With B only, it is anti-B. It is blood group B. With anti-A and anti-B, it is blood group AB. With an O no happen, no agglutination happen, this person doesn't contain any antigens, so his blood group is O. Okay? This when you are making a test for a person to know his blood group. Agglutination happens with anti-A only, positive result. So this is blood group A. Agglutination happens with anti-B only, positive result with anti-B. So this is blood group B. Agglutination happens with anti-A positive and with anti-B positive also. This is blood group AB. No agglutination. This is blood group AO. The risk of blood transfusion. It is very dangerous because many diseases can be transmitted with blood. When incompatible blood with his blood group is transfused to him, this includes symptoms such as severe headache, chest pain, and breathlessness, and lowness, trachycardia, and hypotension, and often end with death. A big dangerous for a body if he has a blood group and you give him a wrong blood group, incompatible blood group. And the viral infection can be transferred like virus AIDS or hepatitis C can take place by blood transfusion only, since it doesn't transfer among couples or from the mother to her fetus. And the AIDS virus also can transfuse with the blood. Blood is subjected to a range of blood tests to make sure that it is free of pathogens, no diseases in it, as viruses and so on. And it must be compatible with the recipient or the receiver blood group. So it is very important to know your blood group to avoid the danger of blood transfusion. Finally, we reach the rhesus factor. The rhesus factor is another antigen. The rhesus factor is another antigen called the rhesus antigen, found on the red blood cells. In addition to rhesus found on the red blood cells, in addition to antigen A and the antigen B, it is another antigen called the antigen of rhesus. Antigen of rhesus also is important to study because it maybe also make some problems if blood donation happen with the wrong rhesus factor. The rhesus may be positive and may be negative, and most people are positive. 85% are positive rhesus. 85% are positive rhesus, and only 15% of people are negative rhesus. So the majority of people are positive in rhesus factor. The rhesus factor, inheritance of rhesus factor antigen is controlled by 3 pairs of genes. 3 pairs of genes located on a pair of chromosomes. 3 pairs of genes يعني ايه مثلا؟ يعني مثلا A, B, C. 3 we have 3 pairs of genes. طيب امتى نقول الشخص ده بوزيتيف ريسس وامتى نقول نيجاتيف ريسس؟ لو عندي بوزيتيف ريسس بيبقى احتماله كبير جدا. يقول لي ايه؟ the presence of any gene or more of these genes repairs dominant state leads to formation of rhesus factor antigen positive and the person become positive rhesus RH positive. لو اي واحد من دول dominant يعني capital letter واحد only one only, so he will be RH positive. RH positive لو one only dominant A capital بس او B capital بس او C capital بس او 2 A capital او 2 B capital اي capital موجود اي dominant gene يبقى انا عندي ده RHA positive. عشان كده البرسنتج في 85% من الناس عندها بوزيتيف ريسك. طب امتى يكون نيجاتيف بقى؟ احتمال واحد فقط. لازم يكون all genes are a negative rhesus عشان يبقى نيجاتيف. عشان يبقى نيجاتيف لازم يكون ايه؟ مفيش غير احتمال واحد، كله يكون ايه؟ ريسيسيف. ده الاحتمال الوحيد. لكن لما اقول ايه كابيتال مثلا سمول بي كابيتال بي كابيتال بي سمول بي سمول سي سمول سي سمول، في واحد بس كابيتال، يبقى بوزيتيف ولا نيجاتيف؟ يبقى ار اتش ايه؟ بوزيتيف. امتى بس يكون نيجاتيف؟ ار اتش يبقى نيجاتيف امتى؟ اونلي لما يكون كل البيرز دي كل الجينز دي ايه؟ نيجاتيف ريسكس. لو واحد فيهم بس بوزيتيف بالتالي يبقى انا عندي البوزيتيف احتماله اكبر بكتير جدا. طيب الريسس فاكتور ده اهميته ايه؟ Importance of rhesus factor. Rhesus factor determination shouldn't be neglected. مش ممكن نهمله. Before blood transfusion لازم نعرفه كويس قبل ما نعمل نقل دم. As with as before marriage to avoid risk of a arising from formation of antibodies for rhesus factor antigens that cause disintegration of red blood cells, destroying of red blood cells. You have to know rhesus factor positive or negative. So some people have blood group O positive and some people have blood group O negative. Some people have blood group A positive and some people have A negative. The meaning of positive and negative, this is the rhesus factor, positive or negative. Is it okay? Okay. Let's continue. If we have rhesus factor, what is the risk of rhesus factor during pregnancy and delivery? During pregnancy and delivery, there will be a big problem if the mother was negative rhesus and the baby or the fetus was positive rhesus. If the mother was negative rhesus, it will be very dangerous for the baby if he has positive rhesus. What happened? The mother by blood identify the positive antigens and they make some antibodies to destroy the blood of the children if he was positive. But the baby, this mixing of blood happens during delivery when the baby go outside. But the antibodies stay in the body of the woman, preparing itself for any positive rhesus, any new pregnancy. If a new baby comes with positive rhesus during pregnancy, the antibodies found in the body of the woman will kill the new child, if he was positive rhesus. So to avoid this danger, to prevent the preventive measure that we can do in this case of discovering this difference before delivery of the first baby is injection of the mother with protective serum. If you want to avoid the dangers on the second baby, we have to inject the mother with a serum to before passing 72 hours to prevent the formation of antibodies in the body of the mother, so the mother doesn't kill the blood of the mother, the antibodies doesn't be formed, don't be formed, and the mother doesn't kill the baby, the new baby. The serum disintegrates the blood containing Rh positive that leaked from the blood of the fetus mother to blood before enhancing mother immune system for making antibodies. So if the mother was negative rhesus, we have to take care. We have to take care before pregnancy. If the mother was negative rhesus and the father was positive, it will be very dangerous for the baby, not the first baby, but the second baby. This case only happened when the woman was negative rhesus and the man was positive, and the baby, the first baby was positive. It will be a big problem for the second baby, not the first baby. It will be problem for the second baby because the antibodies formed in the body of the mother, which can kill the second baby during pregnancy. This was the rhesus factor. You need to read it more to understand it more. You need to read it more and more. But to avoid the risk of arising formation of antibodies against the antigens of the rhesus factor that cause the disintegration of the red blood cells, the determination of rhesus factor shouldn't be neglected before blood transfusion process or before marriage. We have to know the rhesus factor, is it positive or a negative. We come to the end of the session. Next session, inshallah, we will discuss complementary genes. In the next session, inshallah, we will discuss complementary genes. I will send you an exam for the first term, inshallah. I wish you try to solve it, to not forget the first term. I will send you an exam to try training, solving an exam about the first term. Thank you and goodbye. Until we meet again, thank you and goodbye. Assalamu alaikum wa rahmatullah.
Alaikum assalam.